Many of us are familiar with the standard treatments for cancer, like chemotherapy and radiation. And while those are still widely used, scientists have been creating much more personalized forms of care that are, in many cases, much more effective than the usual therapies. One that's been in the works for decades is called chimeric antigen receptor T-cell therapy, more widely known as CAR T-cell therapy. Dr. Michel Sadelain, a pioneer of this research, says it's used to treat some types of blood cancers by teaching the body's immune system to fight off the disease. T cells are white blood cells in your immune system that help kill infected or cancerous cells. What this novel treatment does is it genetically modifies these immune cells to recognize specific molecules unique to the invading cancer so that it can more quickly find and kill the tumor.
They are cells, meaning they're alive. And the starting material comes from the patient himself or herself. So we collect these cells, which is very easily done because we all have immune cells, including T cells circulating in blood. So you just need to draw some blood to get T cells from the patient. They are then brought to a laboratory where they are processed to acquire this CAR molecule, and then they are reinfused. So not only is it different because it's a cell, but it's also a medicine that is given only once. Because cells are alive, they can divide, they can multiply, they can persist in the body for days, weeks, and even months, and sometimes years. And their job is to fight the cancer.
Sadelain is the director of the Initiative in Cell Engineering and Therapy at Columbia University. He and 3 other researchers were recently awarded the 2025 Merkin Prize in Biomedical Technology for their roles in pioneering this therapy. Though the treatment was first FDA approved in 2017, Sadelain's been working on CAR T-cell therapy since the 80s when he and other colleagues first developed it.
After obtaining a medical degree and then working at that time on a PhD in immunology, it occurred to me that perhaps we should teach our T cells to do what we would like those T cells to do. And for that, it would have to be a genetic order that we insert into those T cells. So then I had to become a genetic engineer. And so after my PhD in Canada, I went to MIT in Cambridge, Massachusetts, to learn what was the beginnings of the science of gene transfer.
Gene transfer refers to the process of passing on genetic information. We do this naturally between parent and child during reproduction, but in medicine, it occurs when scientists modify living cells, then insert them into humans. In Sadelain's case, he used it to see if he could alter T cells to attack specific biomarkers found in cancer.
And once it started succeeding in these T cells, these cells of the immune system, we could really study these molecules, receptors that you would have to create to reprogram, repurpose, and target these T cells to fight cancers. And thanks to these methods for genetically modifying T cells, we could conduct a number of studies in the laboratory throughout the 90s into the early 2000s.
The team was able to identify a molecule called CD19 that's expressed by various types of leukemia and lymphoma cancers. After years of research, they were able to start testing their first patients in 2007. These patients had refractory leukemia, meaning that the cancer cells had stopped responding to all other treatments.
These therapies are approved once cancers have become resistant to all current or previously known treatments, the so-called refractory disease. So the disease is really bad by then. And in all the first trials that started testing these CAR T cells, those were patients who were going to die from their disease within months or maybe not much more than a year or so. So we're talking about assessing responses in patients who didn't have any hope anymore until the CAR T cells came around.
To this day, CAR T cells still aren't approved to be a first-line defense against cancer. The handful of approved products are still only allowed to be used for relapsed or refractory cancers.
The way new medicines are evaluated in the US is by starting with clinical trials that first enroll patients who have already received existing approved treatments, that's called the standard of care, and who are now failing with these treatments. So by definition, they have already received one or very often multiple treatments, and that means that their disease is also really advanced. So that's the starting point for clinical trials, and therefore, when these CAR T cells were approved, they were approved for those patients.
However, Sadelain believes it's only a matter of time before more patients will be able to utilize this therapy.
But that requires additional clinical studies where you have to compare this treatment to others that are already in use to be certain that it is better to give the treatment earlier. And this is happening, but it takes time.
Time and proof that this treatment is effective. Thankfully, Sadolin says CAR T-cell therapy has already been shown to cause significant improvements for certain types of cancer.
The first metric is going into what's called complete remission, which is the cancer becoming undetectable. The doctors try to detect it using vast different measurements. We don't call that a cure. We call that a complete remission because we know that there's maybe a few cancer cells left. It's just that we can't find them. So then you have to wait for a long time and see if the cancer comes back. So just to give you some examples, in refractory acute lymphoblastic leukemia, about 80% of patients will go into complete remission. And it seems like at least a third and close to half, perhaps, will have at least a very long complete remission. And after years, at that point, you can call it a cure. In forms of lymphoma, the durable complete remissions concerns perhaps even more patients. 60% of those that go into a complete remission may still be disease-free after many years.
Again, this is against a disease that's become completely resistant to chemotherapy and radiation.
Patients have undergone a number of these procedures and received a number of different treatments that no longer worked or perhaps never did work for those patients. A big difference is that the chemotherapy is kind of a poison that kills lots of cells. The goal is to kill cancer cells, but it does have numerous side effects, I think, that most people are familiar with. Whereas these CAR T cells are part of what is called precision medicine. They are targeted, like a missile that's targeted to recognize very specifically its target. And so this is why they are so attractive, because they can be efficacious while minimizing side effects.
However, that's not to say that CAR T-cell therapy is without any of its own side effects. The treatment can induce severe and life-threatening toxicities like cytokine release syndrome, which happens when the immune system releases too many cytokines into the bloodstream. The treatment can also damage the nervous system and cause confusion, seizures, and paralysis. Rutgers Cancer Institute estimates that up to 90% of patients experience symptoms, but for most it feels like a bad case of flu and resolves in a week. Within that short time, it's possible that as they're experiencing negative side effects, the therapy is already causing cancer regression.
Meaning there are fewer and fewer cancer cells. Now, how long it takes to eliminate every cancer cell in those instances where it's successful is a bit harder to tell. It may be within a few weeks. For some forms of leukemia, we suspect, but we're not sure, that it may take a little longer in some other cancers. There's much more to be learned.
Which isn't a surprising statement, given that Sadolin's been researching this therapy for the last several decades. There's always more tests to run and data to gather, despite his recent award recognition.
The broader public becomes interested in research once it starts reaching a lot of people. We estimate today that about 45,000 and probably more patients have received these cells. So I guess society is starting to take note. But the reality is that the research starts long before. And it starts in research centers, usually in medical schools and in universities. And it takes a while until you think you got it right and you can go to clinical trials, which we had to conduct ourselves at our medical centers. As all recipients of this prize today, my co-recipients, Isabelle Riviere, Carl June, and Bruce Levine did. If you were going to bring this to the clinic, you had to do it yourself because there was no industry at the time that was willing or interested in doing so. So piecing all of this together takes quite a few years.
You can find more information about Dr. Michel Sadolin and all of our guests on our website, ingoodhealthpodcast.org. For more behind the scenes, follow In Good Health Pod on Facebook, Instagram, and X. Our writer-producer is Kristen Farah. Our production manager is Jason Dickey. I'm Greg Johnson.